We can start now.
Okay. First of all, I want to introduce our presenter today, Dr. Sara, and she is from UCL, assistant professor, and she is going to give us an interesting talk about microelectronics and the microsystems for real-time biosensing devices. And she is a very, I'm expecting a very interesting talk, and it's our pleasure actually and our honor to host her talk here in Nile University. And the mic is yours, Sara, you can start. And thank you again, actually, for accepting the talk.
Thank you, Ahmed, for the kind introduction. And it's my pleasure actually to be here. Thank you for the invitation. I'll try to share my screen now. Ahmed, can you confirm if you can see my screen?
Yes, we can see it.
In full screen mode now?
Yes.
Yeah, wonderful. Thank you. So as Ahmed mentioned, the title of the presentation I prepared today is Microelectronics and Microsystems for Real-Time Biosensing Devices. Just to give you a bit of introduction into the topic and a bit of motivation as well. So I assume the background of most of us is in microelectronics, and I just want to remind everybody that the rapid progress of microelectronics and semiconductor devices, semiconductor technology over the past 60 years or so has revolutionized the cost, the rate, and the frequency by which we receive information about the outside world through advances in devices, smartphones, television sets, radio systems, and so on. However, the way we receive the information about the inside world, our own body, has remained almost the same over decades, that is through a blood test, if and when prescribed by our doctor. As you know, the result of the blood test is unlikely to be ready within a few hours. Normally it takes a few days, and it normally only provides a snapshot into the health of our body and the state of our body, and can only pick up highly atypical clinical conditions. So a technology that can provide real-time information about a variety of biomolecules, hormones, biomarkers in our body has the potential to revolutionize the way we receive information about our own body, and through that improve healthcare and benefit the patients. It's not only real-time measurement that matters, there is also the need for continuous monitoring of some biomolecules and biomarkers. In fact, continuous monitoring is still an unmet clinical need, except actually for glucose monitoring devices for diabetes. But in addition to glucose, there are other biomolecules, other hormones and biomarkers that would benefit from continuous monitoring. Examples are hormones such as fertility hormones, cortisol, stress hormones, salivary biomarkers such as salivary pH, and so on. So there are also other things that would benefit from continuous monitoring. For example, if we manage to continuously monitor the metabolism of pharmaceutical drugs in the body, we then can have the means to actually personalize the dosage of the therapy, the dosage of that pharmaceutical drug according to the individual metabolism of the drug in individual person. Well, such a tool that can allow continuous monitoring can also be very valuable for other avenues of research, can help medical and clinical scientists to develop to explore new avenues of research. For example, they can use it to explore new biomarkers of health and diseases. So I hope I've convinced you that real-time measurement of some biomolecules and biomarkers and their continuous monitoring has great potentials, and most of this is still unmet is an unmet need in clinical world. So such a sensing system. So we're talking about sensing, real-time sensing, continuous sensing. So such a sensing system would composed of a few different elements. So depending on what analyte we would like to measure, could be a cell, a protein, a small biomolecule, RNA, DNA, etc. Depending on the type of analyte, we will need a receptor element that can couple to this analyte in a specific way, in a selective way. There are various types of receptors depending on the analyte. Could be a cell, nucleic acid, antibody, or enzyme. And then a transducer would then convert the matching event between the analyte and the receptor into an analytically useful signal that can be processed and displayed. So there are various types of transducers. There are electrochemical transducers, optical, thermal, and piezoelectric ones, among others. So the electrochemical ones have the benefit of providing a real-time measurement, and they're also provide the sensitive, and they're normally also highly sensitive and specific. They can be made in a way that they're highly sensitive and specific, and it's also the focus of our research. So we mostly work with electrochemical transducers. So an electrochemical transducer, if you remember from high school chemistry, is very similar to an electrochemical cell. Typically, there is either 2 or 3 or more electrodes. In an electrochemical cell, here we have an example in this cartoon of 3 electrodes. There are working electrode, reference, and counter electrodes. And there are various ways that we can actuate and sense a signal from these 3 electrodes. So normally an electrochemical reaction happens in the vicinity of the working electrode. So we either actuate the sensor by applying a voltage or a current. The current or the voltage could be fixed or variable. And there are different permutations of this, as you can imagine. I just included the 3 of electrochemical techniques here just to show you the different techniques that you can use. There are some common techniques, for example, applying a fixed voltage between the working and reference to promote a redox, to promote a specific reaction at the working electrode, and then measuring the current of the working electrode. And this current is ideally linearly dependent on the concentration of the analyte of your interest. So you could also measure the voltage. Well, the choice of the electrochemical technique depends on the type of analyte you want to measure, type of electrode, type of receptor that you have, and a few other parameters, as you can imagine. So in the next 15 or 20 minutes, I would like to discuss. I would like to actually present a few results from our research over the past years in line with devices and technologies for real-time and continuous to provide real-time and continuous measurement. I'll start with a system that we designed a few years ago. So we wanted here to show the feasibility of a subcutaneous device for measuring a panel of biomolecules. We actually wanted to see whether we can actually develop such a technology. So that's the idea. So the device was designed to be implanted under the skin, to be in touch with the interstitial fluid, not blood, to measure from the interstitial fluid. So we wanted to measure a panel of biomolecules, small biomolecules, including glucose, lactate, ATP, and so on, through some electrochemical transducers that were embedded on the device. And then we wanted to send this information wirelessly to a flexible patch, which was located on top of the device. So there was data telemetry, wireless data transmission, and power delivery to this device. The device was fully batteryless. And the patch on top of the device would do some noise filtering and so on, and then transfer the data via Bluetooth to a more powerful device, such as a smartphone or a tablet for extra processing and display. So the implantable part of this system had three main parts, three main components. There was an array of electrochemical transducers to measure different biomolecules. So each of these electrodes needed to be made specific to one of these biomolecules. So we needed different types of receptor elements on each of them. Then there was an instrumentation, an integrated circuit to actually control and read out the sensor array, and an inductor for both power and data telemetry, power transmission and data telemetry. So as you can imagine, to make this device compact, both the electrochemical array and the circuit side, everything electronics had to be compact and integrated. And I would so that's the image of the final device we developed. And what I'd like to focus on is the design of the integrated circuit here, which was actually the brain of the system. So here is the block diagram of it. So there are lots of blocks in green and yellow that represent analog and digital blocks. It was a mixed signal integrated circuit fabricated in 180 nanometer technology. So from one side, the right side in the schematic, it could interface an array of 5 different sensors. You can, as shown here, with 5 different working electrodes. And then we also needed to measure the pH and the temperature of the environment to calibrate this electrochemical sensor. So hence a pH sensor and the temperature sensor on the sensor array as well. So these could all be interfaced, as in, so focusing first on the 5 working electrodes. We could either measure the current of these working electrodes through a transimpedance amplifier followed by a sigma delta modulator, or we could keep them prepared for measurement through applying various different waveforms that are required. I mean, that tree of electrochemical techniques that we didn't go through, for different type of measurements in that tree we need different voltages to be applied to the sensor, and for the sensor to be kept at that voltages for some time before we can actually start reading from the sensor. So there were various elements here. It was the most advanced instrumentation circuit at the time, because not only it could read out the sensor, but it could keep them prepared for the measurement. And all the signals, all the waveforms were produced based on a single digital to analog converter, followed by different sample and hold circuits that were controlled internally through some digital control units. So from one side, as I said, the device, the integrated circuit was connected to the sensor array, and from the other side was connected to another integrated circuit for that would transduce the received AC power to DC, so would give just a DC voltage of 1.8 volt to this IC, and also take care of modulating the inductive link for bidirectional data and data communication. So there was data going from the implant outside and commands being received from the patch into the implant. So thanks to clock gating, thanks to power gating and changing the clock according to the operation of the device, we managed to design this such that it only consumes less than 1 milliwatt power, and it could comfortably be powered through the inductive link. However, we had some problems powering this device through the inductive link, because in what I showed you here, we used a single inductor for both data and power telemetry, and the bit error rate wasn't great. So that's why we moved to another design where we used an antenna we used actually 2 channels, an antenna for transferring the data and a coil to receive power. So we used 2 different frequency channels for the 2 to actually improve the bit error rate. And that's actually the block diagram of the second implant and second system based on the same integrated instrumentation circuit that we designed. So as I mentioned, so we had an antenna and a coil. On the implant side, we had all the different circuits to convert AC to DC, and the instrumentation circuit again to work with the sensor array. And we also had, we used, I think, on-off keying for transferring the data back to the external unit. So on the external unit, which was designed on a PCB, we had a driver to send the power to the implant coil. We also had on the receiving side of the data, we had the transceiver side, we had noise filter, we had filters, we had some comparators and some other circuits to actually extract the data and transfer it, process it and transfer it directly to the display unit. So all the processing was done on this external PCB unit. So we managed to test this device in vivo. We implanted it in a lab rodent, and I just show a cartoon of the experimental unit here. So the device was implanted in the stomach region of the rodent, and it was powered through the external unit that I just described in the previous slide, which was located in the bottom of the cage. And the device itself, like before, it had three main units. It had the implant had the electrochemical transducer, the integrated circuit, plus the power and data management circuit, and the antenna and the coil. So we had more stacking here. We had 3 stacks, as you can see on the top right. And what you see on the bottom left is the final device that we actually then implanted in the rodent. It was encapsulated in a biocompatible coating. And finally, we took some measurements from this device. So we injected the animal with a dose of paracetamol and took both measurements from the device and from blood extracted from the animal at the same time. And we saw a good matching between the measurements we took from the device and the measurements of with the external sensor, with the blood samples. Okay, so moving on. So the devices that I showed you so far, they use 2 separate substrates to fabricate the sensor array and the electronic side. So that's what we call a 2-chip biosensor. We have the instrumentation and the sensor on 2 separate chips, and these are 2 separate chips or 2 separate substrates, and these are connected using wires. As you can imagine, using interconnects, wires that are long going from one substrate to another would damage the signal to noise ratio of the overall sensor, overall sensing system, and it will also limit the scalability of the device. So the reason we were limited to 4 or 5 sensors was because connecting from one to the other was one of the most challenging steps that I didn't go through, one of the most challenging steps in the full integration of the device. So what we suggest is to actually put both the sensor and the instrumentation on the same substrate and monolithically actually integrate these sensors onto the semiconductor devices on silicon. So we're focusing here in CMOS technology and trying to integrate the sensor elements, working electroreference and counter electrodes on the same silicon CMOS device. As you can imagine, the benefits of integration or scalability, we can scale the device to have so much more number of electrodes. It will also allow us to do in-pixel processing, amplification in pixel, and by pixel I mean a single transducer and all the associated electronics. It will also have some other benefits. So scalability and improve SNR are the immediate benefits, but it can also allow us to reduce, because through integration we can also achieve miniaturization, and through that we can also we can also do the same type of sensing with a much smaller volume of the biofluid, which might be costly or difficult to obtain. Well, the other benefits that you can imagine are we might be able to measure several different biomolecules at the same time. But there are some challenges in developing this technology, actually, which is what has been keeping us busy over the past year or so, and also it's going to keep us busy also in the next few years. So integration of the sensor and the electronics comes with some challenges. The first one of them is that so the intrinsic metal layers that we have on the CMOS technology are normally aluminum, which are highly electroactive and not really suitable for sensing applications. So the very first step to actually build your sensor on top of a CMOS device is to cover that aluminum with a suitable material. Could be an inert material, an inert metal such as gold, platinum, and so on. So we are not the only people who are looking into integrating the sensors into the electronic devices and semiconductor. There are other examples in literature. So I've just included 3 examples of these in this slide. On the left, you see a device with 4 sensors. Each of these sensor pixels have a working electrode reference and a counter electrode. The device was developed for gas sensing. On the middle, you see, again, some working electrode reference and counter electrodes on chip, covered with gold for DNA analysis. And on the right, you see an array of ion-sensitive field effect transistors, which is basically, which are intrinsic PMOS and NMOSs on the semiconductor technology with the passivation layer on top of the gate that makes them pH sensitive. So this device, on the one on the right, was developed towards ion imaging by depositing yet more layers that makes the device sensitive to different ions such as sodium, chloride, and so on. So depending on what we want to measure and depending on the type of our sensor, our electrode may need to have a direct contact to the biofluid, to the gas, to the environment actually that we want to measure from. The ISFAD array on the right really doesn't need a direct metallic contact to the biofluid and is covered with insulating materials. But the 2 examples on the left, they do need a direct contact, hence being covered with gold. And there are various methods we can cover these aluminum layers with gold or other inert metals. So we could use lithography in the clean room, which gives us an accurate result, but it's quite expensive. Or we could use electrochemical techniques on the bench that do not require lithography, and they rely on electrical selectivity of the pixels. So they're low cost, but they may have high roughnesses and they really need a lot of optimization and tuning. So that's about the very first layer that we want to put on top of our aluminum, but there is another challenge when we miniaturize our sensors to put them on the semiconductor devices. The signal of the sensor also scales down. The signal is normally directly related to the area of our device. So if you make them very small, the signal is also going to be small. So in order to enhance the signal of our electrode, what we can do is to also deposit not only the metallic layer, but also some nanostructures that would enhance the 3-dimensional area of our electrode. And that's actually what we have shown for the first time with platinum nanostructures. So we developed this integrated circuit with electronics that I'll show you in the next slide. And we wanted to see whether it is feasible to develop platinum nanostructure directly on the sensor using bench techniques, using electrochemical techniques to selectively only deposit these platinum nanostructures on one electrode and not the rest of them. And we actually managed to do this. So what you see is in the middle is SEM image of the electrode that we selectively deposited platinum nanostructures on. And we actually use this sensor, we use this electrode to measure hydrogen peroxide, which is an important byproduct of enzymatic sensing techniques that we normally use for small biomolecules such as glucose, lactate, and so on. So we showed that with these platinum nanostructure devices, we can enhance the signal of the sensor by 2 orders of magnitude. So that is shown here. So the sensitivity of the sensor, which is the slope of the current versus concentration line, has increased by 2 orders of magnitude through introduction of these platinum nanostructures to the surface. But we also had an issue here. Introducing these nanostructures increased the background current of the sensor a lot. So as you can see here, the background current of the sensor is in the range of is pretty much in the range of the signal we receive from it. And the challenge of this background current is that it means that our analog to digital converter at the end of the readout circuit has to have a high number of bits to be able to resolve the signal on top that sits on top of this background current. So to eliminate this issue and to relieve the constraints on the ADC, we developed a differential sensing technique based on not only the electronics instrumentation, but also on designing the sensor differently, such that we can eliminate the background current before the ADC. And it's actually a simple technique. So we thought, okay, so if we have, instead of 1 working electrode, if we have 2 of them, and if we deposit our nanostructures on both of them, but only make 1 of them sensitive to the analyte through depositing the receptor elements on only 1 of them. Here is the electrode on the top. And then if we then measure the signal through these 2 electrodes and subtract the signal before giving them to the ADC, then we'll probably eliminate the background current. And we actually implemented this, and I went all the way to actually prove this idea. And we managed to show that with this differential sensing technique, we can reduce the background current by up to 95%. And not only that, we can also reduce the drift, actually not reduce, but address the drift of the sensor and compensate for that by up to 60%. So additionally, this instrumentation circuit had one of the highest dynamic ranges among the circuits. When we published this, it was 170 dB. Cross scale was from picoampere range all the way to tens of microampere range that it could measure. Right, so we continued to improve on both the circuit and our integrated sensors. So that's the 2nd generation of a monolithically integrated sensor on chip. So here, instead of 1 sensor array that we had on the previous chip, we have a few of them. We have 4 of them here. And we also incorporated ISFETs for pH sensing on each pixel. We also on the circuit size, we have more powerful circuits. That means we can accommodate for more electrochemical techniques on that tree of electrochemical techniques I showed you in the very first slide. And we are actually trying to deposit various types of nanostructures on top of these electrodes, and the results are emerging. Thanks to the support of UK Research Council, we are also continuing to increase the number of arrays here and also incorporate various types of calibration techniques, not only measurement, but calibration techniques as well on chip, so in each pixel. So hopefully the result of this will also be published in the next few years. Right, so I would like to, if I have time, Ahmed, I think you told me 45 minutes, right?
Yes, but it's okay, you can keep going.
Okay, yeah. I think 10 or 15 more minutes would be enough.
That's fine.
Yeah, so I would just like to change gears slightly. So far I've been talking about sensors and electronics. But over the past 3 years, I've had the pleasure of working with within a department at University College London that was located in a hospital, in an orthopedic hospital. So we had the pleasure of having breakfast and lunch with clinicians and surgeons and discussing research and thinking about common interest and so on. And we actually reached an interesting point. So we discussed pain assessment techniques. Just to give you some motivation, again, an introduction in case you don't know about this already. So pain is a pretty vital function in our body. It protects our body actually by giving like enough alert about a stimuli, a harmful stimuli, and initiating fight or flight response. So, but if the pain is not managed properly, it can lead to chronic pain. And if it is not managed properly, it can be physically, it can be draining, and psychologically, it can be distressing. So there is a huge bulk of research on methods for pain management, but the very starting point of pain management is assessing pain and seeing how pain management has affected the pain. And at the moment, the methods for pain assessment mostly rely on self-reporting. So if a physician wants to see how much pain you're in, they will simply ask you, give you one of these scales probably, and ask you to mark your pain level. And this is what we mean by self-reporting. So although this might be effective for majority of adults, it's not really working for infants, those who may not be able to self-report, for example, those who have cognitive impairments, mental impairments, or those who are under induced coma or anesthesia that cannot really self-report. For those groups of people, there are behavioral or physiological based methods for assessing their pain, but those rely mostly on the expertise of the healthcare professional, which limits their reliability. There are also physiological techniques, as I just mentioned, but they have also limited reliability because they're not only a function of pain, they could change as a function of other conditions as well. So there is a consensus in researches in pain that we need to develop more objective ways of assessing pain. And if we manage to develop such a technique, such an objective measure of pain, then we would be able to actually tune our pain management techniques better in a more effective way. So in search of a more objective pain assessment technique, we did two things actually. We actually did a bit of literature review and found some salivary-based biomolecules and biomarkers which are related to pain. And we focused on saliva. So we tried to see whether the concentration of these biomolecules and biomarkers in saliva changes with pain. And the reason we focused on saliva was because we wanted to have a non-painful method of assessing pain, if that makes sense. So the different biofluids that we could access on the body, sweat, for example, but we selected saliva because of its abundance and its ease of sampling, and also it's been shown in literature that concentration of proteins and some biomolecules in saliva correlates with their blood concentration. So here what we did was to obtain some ethics approval and recruited healthy volunteers, 20 healthy volunteers, and we induced pain experimentally on them by asking them to submerge their arm in an ice bath. Then we took their saliva samples every 10 minutes before and after this. And then we focused on a few biomolecules here. So we found that cortisol, through our literature review, we found that cortisol is a biomarker of physiological stress. So that's why we were interested in cortisol. But glutamate was also one of our targets analyzed here to measure, because it's it's involved in the pathway of pain. So we analyzed these two biomolecules, glutamate and cortisol, in all the samples that we obtained. We observed that for cortisol there is a statistically significant change between 10 to 30 minutes after the induction of pain. So that CPT stands for the cold pressor task. It is simply the painful task that we use to induce pain. And for glutamate, we saw some variations, but none of them were really statistically significant. We think that the reason we didn't observe anything might be because we missed it, that the sampling time was just too large. 10-minute sampling time was too large. So we obtained more clinical approvals to repeat this study using other pain induction methods, using a smaller sampling time. So again, we weren't the only people who were interested in developing objective techniques for assessing pain based on salivary biomarkers. We did a systematic review of literature, which took my RA researcher over a year actually to conduct, because there were just so many papers. We started with 1,800 papers, and we applied some inclusion-exclusion criteria. We ended up with about 40 papers. And we made a list of all biomolecules that were shown in these papers that have a variation with pain. And the list roughly contains 10 or so molecules. And we're going to submit this work for publication soon, hopefully. But we are looking into other molecules in the list as well, trying to tune our experiments to see the effect of pain in the other molecules, to see if first we can replicate what we saw in this publication, and second, if there is a scope for using this as an objective measure of pain. So that's on the I would say clinical side of the story. We also looked at so say we managed to find a list of biomolecules that whose salivary concentration changes with pain. So we also looked at potential technologies that would allow us to continuously measure these biomolecules in real time in saliva. There are just a handful of examples actually published in literature. There is no, as far as I know, there is no commercially available intraoral wearable sensor. But there are a few examples in literature that have been published. We looked at, so what I'm showing here are 3 examples. So the one on the top left is a Bluetooth-based a smart mouth guard for measurement of uric acid. The one on the right is a graphene-based device for detection of bacteria. And the last one, the third one, is a wearable sensor for measurement of sodium in saliva. So we looked at the strengths and shortcomings of these devices, and 2 out of 3 they use a battery, which just makes obtaining the clinical approval to run our pilot studies a bit more challenging. We are now discussing clinical approvals is already very challenging. Having a battery there would make it a little bit more challenging. So that is why we want to try having a battery on our wearable device. And the device on the top right, although it's very promising, it's based on measuring the conductance of a tooth tattoo, and measuring the conductance is through an inductive link. And as you may know, such a measurement would be prone to noise and issues due to alignment of the inductors, of the 2 inductors, and the distance of the inductors. So we went on to develop our first prototype of the hardware side of a salivary sensor, or wearable sensor for saliva measurement. And that's the block diagram of the device we developed. It's based on near field communication, and we could just tap it using our tap it using our smartphones. It was based on off-the-shelf components, just 3 IC components, an NFC chip, a microcontroller, and an analog front end. But this was to measure pH. So we started with the easiest biomarker to measure, which is actually, I mean, it's easy to measure, but it's quite challenging to develop a long-lasting sensor for this that could actually work for a few days. So as I said, it's a PCB-based device. You have an image of it here. On one side of the device, we had the components, and on the other side, we had the pH sensor incorporated. So we managed to actually measure the variation in salivary pH over time. So these aren't yet in vivo results. So we took salivary samples every 10 seconds and put it on the sensor side of this. And what we have here is on the vertical axis is what we see actually on our Android interface. So that is basically variations in pH after the consumption of an acidic meal and an acidic drink, showing that actually this simple device works fine. And now we're working to integrate this into a mouth guard in collaboration with dental scientists, periodontists at Eastman Dental Institute in London. Okay, so I'd just like to summarize this talk. I hope I have convinced you that fully integrated, batteryless, and minimal invasive implantable multi-target biosensors are feasible thanks to integrated circuit technology and thanks to electrochemical sensing techniques. And I showed you just one example of how we can co-design our electronics and our sensors to eliminate some non-idealities of the sensor, such as the background current, and at least one technique to develop our single chip analyzer, a chip that has nanostructured electrodes on top and instrumentation in its core. And I also showed you some in-mouth salivary analysis that offers a non-invasive route for obtaining dynamic chemical information related to health conditions. We focused, in terms of technology, we have focused on pH so far, but in terms of applications, we are interested in pain and physiological stress, and we have looked into some relevant biomarkers of these, and research is ongoing actually on all these 3 topics. And thank you for your attention. If you have any questions, I'm very happy to take them.
Thank you very much, Sara, for the very interesting, informative talk. We really appreciate this informative talk. So let's first, if anyone has any questions, could you please raise your hand or just go ahead. I need also to use the chance and welcome Professor Kostas for attending the talk. He's from Greek, and it's our pleasure also to attend the talk. Okay, so in this case, can I ask questions, Sara?
Of course.
So you have talked about wireless power transfer and wireless and battery-less devices. How much? What was the efficiency for the wireless power transfer?
Oh, I actually don't remember the efficiency numbers. I know how much power we managed to transfer. In the device where we had 2 wireless channels for data and power transfer, we managed to transfer 3 milliwatts, but I don't really, I'm sorry, I don't remember how much we transferred on the transmitter side and the efficiency.
Okay.
We can also look at the papers, yeah.
Yeah. Also, one more question. Did you explore the usage of flexible substrate or flexible material?
No, not yet. So we are focusing on very small devices at the moment, millimeter size, and we haven't really explored flexible at this point.
Okay, we have so this is question.
Sorry?
Yeah. No, that's for me. These are the questions. So, if anyone else has any questions? So, okay, in this case, we want to thank you, Sara, for accepting our invitation for the talk, and really thank you. And
my pleasure.
see you in another talk.
Thank you. Have a nice day.
Thank
Bye.
you. Bye.